What do you have up on the screen now that presumably I'm looking at or we are looking at?
Right now, it's just what looks like the Brady Bunch, but we'll put up a presentation shortly.
Okay, so it's just the agenda showing.
Yes.
Yeah. Okay. Thanks.
We don't see the agenda, Maria.
Just a second. We're going to put all that up. Doug, do you want to share, or Gil, are you sharing the presentation or is Doug?
I believe Doug is.
Doug, if you could put that up, just the front, and then I'm going to put myself on mute. I just need to get Oz on and then we can start the call.
It actually looks like Oz is on.
I don't think we can hear him. Oz, I know you can hear us. Can we hear you? You want to say something? Not on mute?
Is JT coming?
Yeah, I'm here, Art.
Oh, there you are.
It looks like Oz and Dave Martin just came in.
I think they've been on. Okay, so I just sent Oz the link again. I hope it works.
Is that Havana in the background, Maria?
It is. I just wish I was at a beach in a tropical location. Okay, let's see what Oz is texting. Why don't we start, I think, and go from there, JT, and hopefully we'll get Oz on. I haven't received it yet. I'm on both my phone and Zoom. I can hear you. Okay.
Great. Okay. Well, thank you, everybody. I'd like to call the meeting to order. This is the emergency meeting of the Board and Application Review Subcommittee for May 15th. Hope everybody is continuing to stay safe and healthy with their families. Maria, will you please call the roll?
Anne-Marie Dulage, you are on mute.
Yes, present.
Isabel Dudon, David Higgins.
Here.
Steve Julesgaard.
Here.
Dave Martin.
Here.
Lauren Miller,
Here.
Adriana Padilla, Joe Panetta,
Here.
Francisco Prieto, okay, Robert Quint,
Here.
Al Rolette,
Here.
Jeff Sheehy,
Here.
Oz Stewart, Jonathan Thomas,
Here.
Art Torres,
Here.
Diane Winiker.
Here.
We have a quorum, and I'm going to mute myself and try and get Oz and Francisco on.
Thank you, Maria. So today is the second of our series of meetings to consider COVID applications from this week's GWG review. In addition, we have a sickle cell project on the agenda for consideration. And at this point, would like to turn the meeting over to Mr. Sheehy.
Thank you, Chairman Thomas. I think, Dr. Sembrano, are you going to take us through this? We're going to start with the sickle cell one today, aren't we?
Yes, that's correct.
Great. You have slides?
Yes.
Please.
So, okay, thank you. So good morning, everyone. We are going to talk about first the sickle cell disease program and the one application that is being considered. So if I could have the next slide, Doug. The sickle cell program is one, as you know, that is in collaboration with the NHLBI to, where possible, co-fund programs in the clinical arena. So that's the late stage preclinical projects or clinical trials. In this particular case, we have an application that is not being co-funded. This would be an application that was not deemed eligible for the NHLBI funding, but is for CIRM. So this would be a CIRM only funding. Next slide, please. So the scoring, and I want to just emphasize, because we are using two different scoring methods that we're going to talk about today. So for this sickle cell program and for our traditional clinical program, we use a scoring scheme of 1, 2, and 3. So a score of 1, meaning it has exceptional merit and warrants funding. A score of 2, meaning it needs improvement, and usually those get revised and come back. And a score of 3, where it's sufficiently flawed that we wouldn't want that to come back for 6 months. Next slide, please. I want to provide an update on the budget status for the sickle cell disease program. So there was an allocation of $30 million that was created and approved by the board for the program. The amount that is requested for today for the one application is $2 million. We have approved awards that have used up $2.2 million out of the $3 million bucket. And so if application is approved today, that would leave us with $25.8 million. I do want to note that out of this $30 million allocation, I'm sure you all remember, we did borrow $4.2 million for the COVID-19 program. That's not reflected in this chart, but I just want to make it clear that that is the case. We are replenishing that amount with recovered funds as we get them. Next slide, please. Okay, so the project that we're talking about today is a therapy that uses expanded cord blood hematopoietic stem cells to treat severe sickle cell disease. And the goal of this project is to complete a phase 1 clinical trial, and the amount requested is $2 million, and the applicant is providing a co-funding amount of about $860,000. Next slide, please. So as just background information on sickle cell disease and the impact of this therapy, as we know, sickle cell disease is one that has a severe impact on patients. There are approximately 100,000 Americans that this disease affects. It is particularly common in those with sub-Saharan African ancestry, and it affects 1 in 365 African American births. Globally, there are over 300,000 babies who are born with sickle cell disease every year. The value proposition of this therapy comes around in the following way. So the only current cure is allogeneic hematopoietic stem cell transplantation. It comes with some risk, but it is curative potentially, but donor availability is very limited. So the proposed therapeutic approach would broaden the donor availability for patients seeking such transplantation. So that is where the value would come in. Why is this a stem cell project? This therapy includes cord blood-derived hematopoietic stem cells. Next slide, please. So this is just a big picture overview of the related projects that exist in our portfolio. So at the very top is the current application and how that would fit in. There are 4 other active awards that are in our clinical portfolio, 2 that are at the late stage preclinical, and then 2 clinical trial projects. Most of these are focused on autologous approaches of gene correction. And then there's 1 that's focused on achieving immune tolerance through mixed chimerism. And so the current application that we're considering is unique among these in that it is an allogeneic approach that uses cord blood and expands it in order to provide a treatment. Next slide, please. This applicant has not received CIRM funding previously, so nothing much to say on that front. And so the last slide on this application, next slide, please, Doug, is the recommendation and scoring from the GWG. So this received a score of 1, meaning exceptional merit and warranting funding. And so this was a unanimous vote by the 15 scientific members of the GWG. The CIRM team also recommends and concurs with the GWG in awarding the applicant $2 million. Mr. Sheehy.
Thank you, Dr. Sembrano. Could I get a motion to either accept the CIRM team recommendation and fund this project, or to reject the CIRM team recommendation and not to fund this project?
This is David in San Diego. I move to do just that.
To accept, great. So do I have a second?
Yes, second.
So, okay, so David Higgins has motioned to accept the recommendation, seconded by Senator Torres. Any discussion amongst members of the board? Questions, etc.?
Jeff, may I ask a question?
Please.
Gil, can you please clarify what this approach would add specifically to the currently FDA-approved approach of hematopoietic stem cells, if I'm correct, which is already approved?
Right. So this is cord blood derived. So there's advantages to using cord blood that include more tolerance for HLA molecule mismatches. There's lower incidence of GVHD. One of the difficulties with cord blood, however, is that often there isn't sufficient in a single unit to treat patients. And so what this brings forward is the ability to expand cord blood units. So there's a small molecule that the team has identified and uses to expand the cord blood in order to create sufficient material for the transplant. And so by virtue of that, it would allow those that might not otherwise be able to receive one, either because there is no good match, to actually be able to pursue the transplant.
Thank you. Very useful.
This is Dave Martin. Is it fair to ask why it's not eligible for the co-funding?
Because I think NHLBI generally is focused on gene therapy approaches, and they felt this one did not meet their bar for a gene therapy approach.
Thank you.
Additional questions or comments? Is there any public comment for this application? If not, Ms. Bonneville, could you please call the roll?
Anne-Marie Dulage,
I agree.
David Higgins,
Yes.
Dave Martin,
Yes.
Lauren Miller,
Yes.
Adriana Padilla, Joe Panetta,
Yes.
Francisco Prieto, Robert Quint, Dr. Quint,
Yes.
thank you. Al Roulette,
Yes.
Jeff Sheehy,
Yes.
Oz Stewart, yes, he's giving me the thumbs up, so I see it, so it's recorded. Jonathan Thomas,
Yes.
Art Torres,
Aye.
Diane Winokur.
Yes.
The motion carries.
What's our total?
12.
Thank you.
Thank you. Dr. Sembrano, or will you be taking us through the COVID applications?
Yes.
Really quickly, I wanted to confirm that Dr. Melmed is on the line, I believe. How about Linda Malkus? I think Linda's on too. I just
Yes,
wanted to
I'm
note.
on. Yes.
Thank you. And I just wanted to note the other board members that had joined. Thank you.
Thank you. So Dr. Sembrano?
Thank you. Next slide, please, Doug. Okay, so just a quick reminder of the COVID-19 program. It is one that we approved recently with a $5 million allocation, and it accepts projects that span from discovery through clinical trials. Next slide, please. And so this next table just shows you the maximum award amounts from 150,000 for discovery projects up to 750,000 for clinical trial projects. The award duration is relatively short, up to a year for all except the clinical trial, which allows up to 2 years for the program. Next slide, please. So despite those award durations and award amount limitations, we do and have set a pretty high bar for a lot of these projects. There was a clear deliverable that was established for each of these types of projects within 6 months of the project initiation. So for clinical trials, for example, we expect that within 6 months, such projects would initiate enrollment and data collection. For the DISC-2 or discovery projects, that they have data for a viable candidate that could progress quickly to the clinic. And so for today, we're only considering DISC-2 and CLIN2 projects. There are no CLIN1 or TRAN projects that are in the pool. Next slide, please. So the recommendations from the GWG, we had 16 applications, I believe, that were submitted, 12 that were eligible and were reviewed. And so out of those, there are 2 that are recommended by the GWG. Both of those are discovery applications. And so the total amount requested for those 2 would be $300,000. And the funds available are $3.9 million. Next slide, please. And so just briefly, the two applications that are recommended that are discovery are numbers 11811, which received a score of 90. And so this application is entitled Repurposing Amino Adamantane Nitrate Compounds to Inhibit SARS-CoV-2 Viral Activity and to Protect the Brain from Viral-Related Damage. So this is a proposal that intends to do a screen of several compounds to identify those that have the potential to protect neurons from viral damage, as well as to inhibit the activity of the SARS-CoV-2 virus. The second project is one that received a score of 85, and it is entitled Development of a Novel PIK5 Kinase Inhibitor for the Treatment of COVID-19. The goal of this project is to determine if a novel PI5 kinase inhibitor can prevent SARS-CoV-2 infection of type 2 alveolar lung cells. And so that's what this project will pursue. And Mr. Sheehy, Doug is going to put up the display of, or the spreadsheet that shows all the other applications as well, which might be just beneficial to give everybody the overview of all the applications. All members should also have a copy of that spreadsheet available in the materials that were provided. So, Mr. Sheehy.
Thank you, Dr. Simbrano. So the way in which we'll proceed is how we typically proceed with these large groups of grants, of applications. So first I'll take a motion to move any application from the unfundable score range into the fundable score range. Then I'll take a motion to move anything not in the recommended range out of the recommended range, and then we'll vote on the recommended and not recommended as a group. So, so first, is there a motion to move any application from the unfunded range, below 85, into the fundable range?
Yes, Mr. Sheehy, I would like to, if I may, move 11823 from the currently not fundable to fundable range.
Okay.
I'll second that.
And
This is Dave.
by Dr. Martin. Do we have, first, might it be helpful to have Dr. Simbrano do a brief review of that application, Chairman Thomas?
Yes, thank you, Mr. Sheehy.
Sure.
Okay, so application 11823, this one is entitled Mesenchymal Stromal Cells for ARDS, COVID positive and negative patients. And so this is a clinical trial proposal that is intended to be an expansion of an existing broader trial that is looking at these MSC cells as a treatment for ARDS, and so, which is acute respiratory distress syndrome. And so with the rise in the crisis of COVID-19, the idea is to mobilize this trial and allow the treatment of COVID-19 patients and determine if this therapeutic approach may also have an impact on such patients. And so this is an application that received a score of 72. There was one member that, a scientific member that scored it at 85 or above, 13 members that scored it below. So I think, you know, some of the reasons as to why this application did not score in the funding range, I think it was unclear and felt that the pivot to COVID-19 may not be adequate, meaning there was the belief that there were potentially only 20 patients that would be part of the COVID-19 project. And so the number of patients, there was also the risk that no patients with COVID-19 would end up being enrolled the way the proposal was laid out. So I think that was a key concern from reviewers. There was, I think they felt that the rationale, the team, largely most other things were fine and appropriate. And I think there were certainly kudos for the team and the PI. I think there was also some concern that there is another trial that is using a commercially prepared product that is underway. And so there may be some competition there. But I think much of it was related to the number of patients. Now, you know, since we published this summary and provided it to the applicant, we did get clarification regarding the COVID-19 patients. And so it has been made clear that this study will include the broader 120 patients that are part of this study and will assess any COVID-19 patients across all the clinical sites, not just the expanded one. So I think that is important new information that was brought to light. So Mr. Sheehy.
Thank you, Dr. Sembrano. Is there discussion amongst board members?
Yes. Yes, can you hear me?
Yes, Dr. Prieto.
Hi. Well,
Yay.
first of all, I wanted to record. Yes, I was having some technical difficulties, although I could hear you and see you all, but and wanted to record my vote as aye on the previous application. But I wanted to ask Dr. Sembrano, wasn't there also a concern that this application was only going to look at patients who had recovered from the acute phase of COVID-19 infection and looking at the pulmonary sequelae rather than perhaps the more urgent, treatment of the acute infection? I thought I saw that.
I don't recall that for this particular application. I do recall it for another one, but I don't.
Oh, okay. I might be confusing
that was the case here.
Okay, thank you.
I'm just curious if you can hear me.
Yes, Oz, we can hear you.
Fantastic.
A double win. Thank you.
Yeah, Dr. Stewart, did you have a question or comment?
No, I've been having trouble connecting, and I was just doing a voice check. Thank you.
Oh, great. Thank you. Is there another?
This is Al Roulette.
Oh, please. Please.
I was the patient advocate that reviewed this particular proposal. And without talking about my score, the concerns that I had were reflected in the comments given by Mr. Sembrano and in the response by the PI, who I believe is on this call. And so I'm foreshadowing my vote, but I was favorably disposed given the response. So thank you, Mr. Sembrano.
Thank you, Mr. Roulette. Is there another additional board comments?
Anne-Marie, I'm sorry, I don't see how I can raise my hands other than in front of the screen. Gil, given your clarification that in fact the actual sample size in this clinical trial is larger than what the reviewers apparently initially understood, do you think the response address most of the comments and potential concerns from the reviewers? And do they actually lift those concerns that in fact the score should be higher?
So I hesitate to speak for the grants working group and exactly what they would believe, but I can tell you that that was a major concern. I think it was unclear whether adequate numbers of patients with COVID-19 would be available in order to get any meaningful output from the trial. But in addition to learning that the number of patients which would be drawn from includes the broader trial, I think it was also made clear that COVID-19 patients have already been enrolled. So I think those two things would certainly have made a difference in the general view of this application. I cannot tell you necessarily whether it would have put it over the funding line.
I understand. Thank you.
Thank you, Dr. Sembrano. And just by the way, if you do want to, it's fine for people to chime in, but if you do want to raise your hand, you can go to the participants window, and they do have the option there, but it's fine for people to chime in. So I don't want to discourage that. Are there other comments or questions from board members?
This is Dave Martin. I would just like to make the comment, although I've been sufficiently fortunate not to take care of a COVID ID patient. The ARDS seems to be, that and CRS seem to be two of the major life-threatening phenomenon in the severely infected and intubated patients. And this is an application that is so far along in terms of having a product candidate in the clinic that happens to be stem cell based and is treating the life-threatening aspect of COVID-19. So I think that this one, for me, is given that the concerns of the working group were at least addressed by the principal investigator, it seems to me one that is right and on target because it's treating the clinical aspect with a novel approach with a group of centers that are well experienced in this particular clinical syndrome associated with this particular infection. So I would certainly encourage us to seriously consider supporting this, particularly since it's so far along, and to get a clinical trial for this price that already has momentum, I think is an important opportunity for us.
Thank you, Dr. Martin. Additional comments or questions from board members?
Mr. Sheehy, just one last point from me. I want to echo what Dr. Martin said, and I'd also make the point that it's piggybacking off of a previously, this is reiteration, previously funded CERM project that it itself had gone through the GWG process favorably. And so I think that having this as an extension of that to further the ball here makes eminent sense.
Thank you, Chairman Thomas. Additional comments, questions? Thank you. Do we have public comment?
I believe, yeah, I think there will be public comment.
This is Dr. Mathey. Does that mean me?
Yes.
Yes, it does, Dr. Mathey.
If you have comments, please.
All right. So I thank you very much for considering this proposal. I won't belabor because it's already been discussed some the responses to the review. I'm sorry that it was not clear in the way we explained the application that this really is a 120 patient trial, and so it's based statistically on the 120 patients, not the proposed 20 additional patients, and it is double-blind, randomized, placebo-controlled. And in terms of the COVID-19 issue, I would like to reassure the review board that that we are doing everything to enroll these patients. In fact, I think I pointed out in the responses that we have enrolled in San Francisco, UCSF and Zuckerberg, 11 of 13 patients with acute respiratory distress syndrome who were COVID positive. And since I submitted that yesterday, we enrolled another patient just yesterday afternoon. So it's 12 of 14. So we are really doing well on the COVID-19 positive ARDS patients. I think I can assure CIRM that will continue, and that is a major supportive part of our program. And I think I won't go through the other responses. I think in interest of time, I will also emphasize that we have a strong track record of enrolling in prior trials in both California and national trials. I indicated that in the response number 8. And I do think actually that it is healthy for us to integrate with academic and industry for these trials. And I have been supportive of, for example, other efforts in the private domain, such as Athersys, at Multi-Stem Therapy in Cleveland, Ohio. They're trying to start a trial, and I think that's very good. We need more than one effort in this area. And so I think that's part of CIRM's mission. And although I'm academically oriented and I have no financial connection to Athersys, I still encourage both efforts in both areas. That does conform with what Dr. Francis Collins has recommended from the NIH. And I would like to emphasize two other points. We are really focused on underserved populations, which was the theme of our alpha stem cell application and funding from CIRM at UCSF. And we fulfilled that commitment by enrolling the vast majority, for example, of our COVID-19 patients with ARDS at San Francisco General Hospital. And University of California, Davis would increase the opportunity to serve and enroll patients with an underserved background. And I laid out the strength of the application, I think, in terms of the investigators at UC Davis with whom I've worked for many years, and have a particularly talented young associate professor, Dr. Calcutt, who was with us at UCSF and moved to a major position at UC Davis in March. So I think we have all that would be needed to do this very well. And there's strong support from the Department of Defense and the NIH. So why don't I stop there so I don't take too much of your time. But if there are any questions, I'd be happy to take them.
Thank you, Dr. Mathey. I think we have another public comment, right?
Yes. It's from phone number 626-340-5600.
So is that me? Hello.
Yes.
Hello. Do you have public comment about this particular award or application?
I have a public comment for DISC 2 COVID-19 11813.
Oh, great, great. And if you don't mind, could you state your name for the person taking?
May I start?
Please.
Hello, Chairman Palmer and members of the board. I'm Ian Hong Shi, a professor from City of Hope. I'm here to ask you to consider funding my application, DISC 2 COVID-19 11813, combating COVID-19 using human TSA-derived NK cells. This proposal received a score of 78, only a few points below the funding line, because multiple reviewers feel that this proposal could yield a significant impact on the field. I ask that you consider funding this study. Today, there are no approved prophylactic or therapeutic options for COVID-19. And more importantly, it's likely that COVID-19 will be with us for the foreseeable future. Even with the future vaccine, patients who are severely affected by this disease will continue to appear. Thus, having treatment options available could be life-saving. NK cells play an important role in regulating the defense against the virally infected cells and the tumor cells. Depletion and exhaustion of NK cells have been shown to cause disease progression and poor clinical outcomes associated with viral infection and cancer. In this regard, immense advancement has been made in cell-based immunotherapy to enhance patients' immune systems through adaptive transfer. We propose to use genetically engineered pluripotent stem cells to make an unlimited supply of NK cells with enhanced killing effects to treat COVID-19. These NK cells could effectively kill virally infected cells and resist virus-induced exhaustion. The reviewers acknowledge that this project is straightforward and streamlined. If successful, could be moved to patients quickly. One potential concern was that an NK treatment could have toxic side effects. But to date, NK therapy for cancer has had minimal toxicity. An added advantage of NK-based immunotherapy is that NK cells don't have the side effects often associated with immunotherapy, such as cytokine storm. Multiple clinical trials have demonstrated that NK cells are safe. The human pluripotent stem cell approach proposed in this study will allow us to generate an effective NK cell therapy for COVID-19. Numerous COVID-19 patients desperately need treatment. We hope that through our research, these patients can be saved to live a normal and fulfilling life. Thank you for your attention. On behalf of COVID-19 patients and their families, I hope you will approve our proposal for funding. Thank you.
Thank you. Senator Torres, I see that you have your hand up.
You're on mute, Art.
So this is not on a on the motion which I support that's before us, but on another application which I sat through as well, where the score.
So Senator Torres, could we wait and get through public comment and get through this?
Oh, okay, sure.
Yeah.
I thought we were done.
No, not quite. Thank you.
Okay.
I have another public comment from 925-818-8797. And could I request that you speak specifically to the motion that we have before us, which is on the application. I can't see the numbers because the way this shows up on my screen, but it's the
11823.
Right. So if you have a comment on a different application, can I please ask that you would hold for that?
Yes, I have a comment on other application, not this one. I can wait.
Okay. Let us go forward with this one. Is there any other public comment on this application? Great. Could we then call the roll on the motion to move this into the fundable range, please?
Yes. Anne-Marie Dulage.
Yes.
Isabel Duran. David Higgins.
Yes.
Steve Fieldsgaard.
Yes.
Dave Martin.
Yes.
Lauren Miller.
Yes.
Adriana Padilla. Joe Panetta.
Yes.
Francisco Prieto.
Aye.
Robert Quint.
Yes.
Al Rolette.
Yes.
Jeff Sheehy.
Yes.
Oz Stewart. Oz, are you on mute? You're on mute. After all that. Thank you.
You saw his thumb?
Jonathan Thomas.
Yes.
Art Torres.
Aye.
Diane Whitaker.
Yes.
Thank you. The motion carries.
Great. So Senator Torres, I think you wanted to make a motion about another application or to comment on another application.
Yes, I just wanted to ask Gilbert regarding application by Dr. Zhang from Cellularity 11857. The reviewers during the examination, we all gave it pretty high marks in the high 80s and 280s. And I was wondering, what were the other scores? Because I can't get that on my screen because the text is too big for transmission.
Sure. So this is application 11857. And so this was a phase 1/2 study of human placental hematopoietic stem cell derived natural killer cells for treatment of adults with COVID-19. So this had a median score of 84. So it's just 1 point below the funding line. There were 5 members that scored basically 85, because that was the highest score given. And then there were 9 members that scored below that, and that would be between 70 and 84.
And can you tell us what the application did?
Sure. So this is a proposal for taking human placental derived hematopoietic stem cells that are cultured into natural killer cells. And so this is a proposal to do a clinical trial to study their impact on COVID-19 patients. So the NK cell population is allogeneic, excuse me, and expanded in culture in order to have sufficient cells to treat patients. I think the concerns related to this application were I think the unknowns about natural killer cells as a therapeutic and the potential relevance for COVID-19. There is some data that suggests that cells such as natural killer cells and T cells become exhausted because of chronic stimulation in COVID-19 patients. And so that is the reason for trying to pursue this and other similar studies. But it is unknown what the effect of the natural killer cells in a COVID-19 patient environment would be. I think some of the fears from working group members is that it could potentially exacerbate the condition, but it is an unknown, right? So it is just a risk.
Well, everything is an unknown right now in this environment, and I think that I don't know whether we should take a risk in seeing whether this process works or not. That was the only issue that I had raised because I was intrigued by the reviewers and what they were hinting at, but also concerned about the exacerbation that many of them said as well. So I'm on the fence. So I think I'm just not going to make a motion and just leave it at that, and unless there are other members of the board that want to opine on this issue.
Other comments, questions from board members on this? Okay, is there any, is there a motion to move any other applications from tier 2 into tier 1? Then is there a motion to move any application from tier 1 into tier 2? Then could I get a motion to approve all the applications in tier 1, including 11823, which we have moved into tier 1?
So moved.
So moved by Senator Torres, second.
I second.
By Dr. Duliege. Is there are there any comments on this? Any board comments and questions on this? Any public comment? I think 925, I think this is probably as good a place as any to chime in, if you'd like to make your
Okay, so can you hear me?
Yes.
Okay, great. Thank you very much. This is Dr. Song Lee calling from UCLA. in bioengineering. So we really appreciate this opportunity to respond to the review. This is regarding the application discovery grant 11838 with a score of 82. It's very close to the cutoff line. And we appreciate the application review committee's insightful comments. And basically, we have unanimous support for the significance and impact of this project from the committee. And the comments on the unique needs, and also this is a promising and novel approach, potentially yet high risk, but high reward project. So that's the comment. The major concern for this comment is the feasibility of the product development and the complexity of this drug delivery system. So this proposal, we're trying to address a major issue for this COVID-19 pandemic. We know that the elderly population is highly susceptible and vulnerable to infection, and there's a high death rate. So that's well known. But there's no solution to this problem. And for these old people, and we know for all the previous infectious disease studies, people know that even vaccine may be much less effective, flu vaccine, other virus vaccine. So what they're trying to propose is to boost the T memory stem cells, which is very important for the long-term memory of the immunity, so which is missing in the elderly population. So we have a drug delivery system proposed, but regarding the feasibility, I would say it's not as complex as people thought. We have three major components, and all of these components actually have been used in other clinical trial approved as products by FDA. So these three components, one is the scaffold material, which is alginate, and there are more than a few dozens clinical trial products approved already. And the second component is this artificial antigen present in cells. And there are at least 6 clinical trials ongoing, including one for T-cell expansion and with the antibody presentation, and expand the T-cells outside body and use that to cure cancer. So that's in clinical trials, several of these examples. And the third component is cytokines. And these cytokines actually are provided by NIH for preclinical trial with large quantity and with good quality. So all of these three components are already have a clear pathway for NIH approval. So we don't think it's a big issue. In addition, as part of our proposed studies, we are going to do a study to examine the effects of each of these components and trying to simplify and see which is necessary and sufficient to induce the T memory stem cells in the elderly. So I think this is feasible, and our team really includes interdisciplinary expertise on stem cell engineering, biomaterials, drug delivery, and virology. And we have all of these materials ready. In drug delivery, we have tested it in T cells, activation and regulation with this drug delivery platform. So we think we can move quickly with what we propose to do and address these concerns on feasibility. I will stop here and be happy to answer any questions.
Thank you, Dr. Lee. Do we have any more public comment on the motion before us?
Jeff, I believe Dr. Zhang has a comment. He might be on mute right now. So I'm scanning. I don't see a hand raised, so I'm not sure. Just one second. Okay, Dr. Casper apparently is who will be speaking, and I don't know. We need a phone number from which they are speaking in order to give them the ability to give a public comment.
Okay.
Dial *9 and you will raise your hand. If you want to make public comment, please dial *9 on your telephone.
So we have two. We have 206-381-0883.
Yeah, hi, that's me. This is Dr. Casper.
Thank you,
Can you
Dr.
hear me?
Casper. Yes.
Yeah, hi. Thank you very much for giving me the opportunity to speak. This is Dr. Corey Casper. I'm the Chief Executive Officer of the Infectious Disease Research Institute and a Professor of Medicine and Infectious Disease at the University of Washington. I'm speaking, I guess, on behalf of Application 857, which is the cellularity trial that was just discussed. This study received what I thought were very favorable reviews, so thank you very much for your careful consideration. As was noted, they were generally favorable, and there were some concerns about the potential safety issues of the NK cell infusion in patients with COVID-19. So ultimately, you're right, it's impossible to know whether these cells will be safe in this clinical condition. It's obviously a novel virus and a novel clinical constellation of symptoms that we're still learning about. But I do think that that it is important to take a chance and to do this investigation. I think that there are appropriate safeguards that have been built into the study in a very careful study design, and I can outline those in a moment. And also, I think that although you can't have any preliminary data for this virus, given that it's so new, the fact that this product has been used in phase 1 studies for cancer in 25 individuals with no significant safety signal, I think is very important. With respect to the clinical trial design, the inclusion criteria for the eligibility of patients in the trial is such that patients only with moderate disease are allowed to enter. So it safeguards against enrolling patients who already have lung injury. And there are additional safeguards that are provided. So one safeguard is that there's a reduced dose of the cells that are provided at the first infusion with close observation before the full dose is provided in the second and third infusion. There's careful monitoring, and it's mandated that the drug, which is currently used in my institution and others for defending against this cytokine release syndrome, these are IL-6 receptor blockers, are available for rescue should patients begin to develop any of these clinical conditions. There's a very careful phase 1 and phase 2 design. The first 3 patients that are enrolled will be evaluated rigorously by a data and safety monitoring board. And then after 11 additional patients are enrolled, they will also be reviewed by the data safety committee. So I think that there are ample opportunities to survey, prevent, and analyze any adverse events that come from this trial. So in sum, I think that we have a product that I think has a biologic plausibility for being very important very unique in its treatment for COVID disease. I think that we have adequate safeguards that have been put into the trial to really assure the safety of the participants. And again, we appreciate the enthusiasm that was voiced by the reviewers.
Thank you. Additional public comment?
We have 2 hands raised. I believe it doesn't, I think it's Song Lee has the hand raised.
Quick comment.
Yeah. Just get public comment?
Yes. Can I make a quick comment?
Quick. I mean, generally, we only do one. You get one shot kind of at public comment, but go ahead.
Okay, 10 seconds. Yeah, I just wanted to add that indeed the review committee pointed out this discovery stage project. It's a challenge, but with the risk for small investment. Indeed, the impact is huge, I think, to address the elderly population's vulnerability to the disease. I hope the board will consider this and be favorable and move this to a category 1.
Okay, thank you. No more public comment, Ms. Bonneville? Okay.
I don't see any.
Okay, so could we call the roll on this motion to fund the applications in tier 1, including the one we added 11823?
Yes. Anne-Marie Dulage? Anne-Marie?
No,
Yes, thank you.
no, no for me. No for me.
Oh, no? Okay.
No, no for me. Yeah. No for me.
David Higgins?
Yes.
Steve Julesgard?
Yes.
Dave Martin?
Yes.
Lauren Miller?
Yes.
Joe Panetta?
Yes.
Francisco Prieto?
Aye.
Robert Quinn?
Yes.
Al Rolette?
Yes.
Jeff Sheehy?
Yes.
Oz Stewart?
Yes.
We heard you. Jonathan Thomas?
Fantastic.
JT?
Yes.
Art Torres?
Aye on all three.
Diane Winokur?
Yes.
Thank you. The motion carries.
Thank you. Now, what we would need is a motion to not fund the remaining applications.
I make this motion.
Okay, made by Dr. Dulage. Is there a second?
Second, this is Oz.
Second by Dr. Stewart. In any board comment or discussion?
Yes, Mr. Chairman, it's Torres again.
Sure.
A question of Gilbert. If we choose to reject all the remaining applications, do any of them have an opportunity to modify or reform their proposal in time for another round that we might have in the very near future?
So the next application deadline is this coming Tuesday. So all applicants have an opportunity to revise and resubmit.
Even if they were rejected today, they can revise and resubmit.
Yes, absolutely.
All right. Thank you, Mr. Chairman.
Okay, thank you, Senator Torres. Any other board comments or questions? Do we have any public comment to this motion? If not, could we call the roll, please?
Yes.
Bonneville?
Anne-Marie Dulage?
Yes.
David Higgins?
Yes.
Steve Julesgard?
Yes.
Dave Martin?
Yes.
Lauren Miller?
Yes.
Joe Panetta?
Yes.
Francisco Prieto?
Aye.
Robert Quint?
Yes.
Al Rolette?
Yes.
Jeff Sheehy?
Yes.
Oz Stewart?
Yes.
Jonathan Thomas?
Yes.
Art Torres?
Aye.
Diane Winokur?
Yes.
Thank you. The motion carries.
Thank you, Ms. Bonneville. Chairman Thomas, this concludes the business of the Application Review Subcommittee.
Thank you very much, Mr. Sheehy, and all participants. We have one other item on the agenda, which is public comment on any topics in general. Do we have any members of the public who would like to say anything at this point? Hearing none, again, want to thank everybody. It's an additional 3 COVID awards. We are definitely doing our part to contribute to this unprecedented worldwide effort to find treatments and therapies for this novel disease. We will have another meeting of the grants working group in a couple of weeks to consider the next round of applications, and we'll reconvene this body shortly thereafter. Please, everybody, you and your families stay safe. And with that, we stand adjourned.
Have a great weekend, everyone.
You too. Ciao, ciao.
Thank you.
Bye.